We saw about the properties of amino acids, right? So I was telling you how to remember this chemical structure of like each amino acids. Okay. So in each amino acid, the backbone, the nitrogen, C alpha atom, and carbonyl carbon, which is making the backbone of amino acid. So this will be remaining constant. Only thing is the R group, the side chain will be varying for each amino acid. Okay. So glycine you will have only hydrogen, alanine you will have methyl group, and valine you will have like a propyl group, and so on. Okay. So we have like a different class of proteins. Okay. So we have like aliphatic or like a hydrophobic amino acids. We have like a charged or polar amino acids. We have and sulfhydryl containing amino acids, right? So we have like a different classes of amino acids, and how to remember we saw the chemical structure we saw in the last class. So I told you to practice yourself to write the chemical diagram of each amino acid. Okay. So you have like arginine, lysine, and histidine, right? So all of you are familiar with amino acid diagram. Nisi Prabhagar,
Yes, sir.
can you draw the chemical diagram of all the 20 amino acids?
Yes, sir.
Yes, sir.
Yes.
Without seeing the textbook, can you draw?
Yes.
Hmm.
Maximum amino acid I can draw.
Which amino acid?
Sir, non-polar or polar I can draw, but I don't remember the cyclic amino acid. Like aromatic amino acid, I don't remember the structure.
Aromatic amino acid you don't know.
Yes, sir.
Why?
I don't remember it.
What?
I don't remember it, sir.
So what are the aromatic amino acid? Do you remember?
Yes, sir. Tryptophan, phenylalanine, and tyrosine.
Okay. So what's the problem with the chemical diagram? Tyrosine and phenylalanine will share same structure, same chemical diagram, except the tyrosine will have hydroxyl group.
Yes, sir.
Okay. So you will have like benzene attached to CH2, okay, and then the backbone. Okay. So
Okay.
this is the common thing. Phenylalanine, you will have like phenyl group, CH2, and then CH, then backbone. Okay, see the double bond O minus, then NH. All right. So this is
Yes.
phenylalanine. So in para position of like phenylalanine, you will have the hydroxyl group. Okay, that is tyrosine. And in tryptophan, you will have like a two rings, 6-membered ring and 5-membered ring. The 6-membered ring is a just benzene. Okay, and then it is attached with the 5-membered ring. Now, 5-membered ring, you will have nitro group, that's all, NH group, which is together attached with CH2, and you will have like a backbone. Okay,
Okay, sir.
so all this chemical diagram it is available everywhere. Just go through and refer it. All right. So you should learn how to draw the chemical diagram of all the amino acids. So do you know what is the chemical diagram of threonine?
Threonine.
Hmm.
Yes, sir.
Yeah. How it
R
is?
group is CH3 and OH.
Okay, that's good. Okay. What about Madhavi? Do you know the chemical diagram of all the amino acids?
Yes, sir, but sometimes I get confused with tryptophan histidine, sir.
Why?
Others I know, sir.
Why you are getting confused?
Yes, sir, the ring structure sometimes I.
Histidine you will have like only 5-membered ring.
Yes, sir.
Okay.
Okay, sir.
And you will have like NH group and nitro group, yeah, nitrogen in histidine.
Okay, okay, sir.
Whereas in tryptophan, you will have like 6-membered ring and 5-membered ring. That's all. In 6-membered ring, nothing is there. In 5-membered ring, only one nitrogen will be there.
Okay, sir.
Right? So we were confusing. It's like a totally two different chemical diagram. Tryptophan, you will have two rings. Whereas in histidine, you will have only one ring. Okay.
Okay, sir. Okay, sir.
All right. Okay. So you have to practice. Now I also asked you to find out the chemical diagram for all the bases. Adenine, guanine, right? ATGC. All the 4 bases. I asked you to practice. So did you do that? Or Madangi?
Yes, sir. Bases, I know, sir.
You know the bases.
Yes, sir.
Okay. So Subhas, do you know
Yes, sir.
all the base chemical diagram?
Yes, sir.
Okay. So, so you have to practice, otherwise there is no chance. All right. So nowadays I cannot come and check you whether you learned or not. I cannot check. There is no facility, right? So even I am asking you to write on a test. So in my absence, you are writing. So it's not a guarantee that you are really learning or you are just copying from your textbook or whatever it is. All right. So I'm little bit afraid. Okay. So whether the quality of like teaching is going degraded or not, I don't know. Like this is like being compromised. So you are sitting far away and I am in another corner, and there is no way that we have like interact in person, and no way that I can like check whether you really learned or not, right? So the condition is getting like very serious and being like worse. Okay, so you have to be like very careful. Okay, you should not take risk by going outside and roaming around, right? It's like a difficult period, and during this period, your education is being spoiled. That is my like very another concern. Okay, so whatever it is, you should take this time for proper learning, right? Otherwise, there is no way. Okay. Okay, so you guys seeing my screen, right?
Yes, sir.
Yes, sir.
Okay. So we are going to see the concept of protein structure. Okay. So, so anybody can guess what is this structure? It's looking like a murku, right? Munya, what is this one? Or jalebi? Come on, yeah.
Sir, first one is secondary structure.
First one is secondary structure. Okay. You know,
Yes, sir. Second is like quaternary structure.
huh?
Second is like quaternary structure, like group of secondary structures and all.
Okay. You are saying this is secondary structure and this is a coordinate structure.
Yes, sir.
Okay. Anybody else? I'm asking whether you know the protein, name of the protein.
Sir, is it hemoglobin?
Hmm.
Second one, I think, is hemoglobin.
So this is hemoglobin. Which is hemoglobin? This is hemoglobin or this is hemoglobin?
The second one, sir.
So this is hemoglobin.
Yeah.
What about this?
Sir, first one is looking like simplest amino acid, that is glycine, and I think I am wrong.
What you are saying?
Sir, I thought it is that's, glycine structure, but I'm wrong. I think I'm wrong.
You are saying this is glycine.
Uh, no, sir. It's a protein structure, complete protein structure.
Complete protein it is having glycine.
No, sir.
Ah. So this is like a polypeptide, okay? So this is a polypeptide, is also like a polypeptide, okay? So somebody told, I don't know, Madhavi or somebody told this is like a hemoglobin, okay? So this is a hemoglobin which is containing like 4 chains, okay? So this is one of the chain from this, or you can say it is a myoglobin, okay? So myoglobin it is having only one polypeptide chain, whereas hemoglobin it will have like 4 polypeptide chain, right? Okay. So how this polypeptide is getting? How we are getting the polypeptide? Just by combination of like different amino acids. Okay. So you have like 2 different amino acids. So when you combine these 2 amino acids, you will get a dipeptide bond by losing some water molecule. Okay. So by losing some molecule, there is a condensation reaction between these 2 amino acids. So the amino group of one of the amino acid and the carboxyl group of another amino acid is getting condensed, and that's why you are getting like a C double bond O, a single bond NH. Okay, so you can see here like single bond NH. So this hydrogen and this hydroxyl group will be like condensed and it will be released as a water molecule. Okay, so resulting you are getting a dipeptide bond. Okay, so this dipeptide bond is like a planar and rigid structure. So like a very important feature in a like polypeptide. Okay, so we will see that in detail in following slides. All right. So when you have like a condensation reaction and loss of water molecule and you have like a dipeptide bond, so similarly several amino acids will condense together and it will make a length of like polypeptide. Okay, so you can have like a n number of amino acid being condensed and you can make any length of polypeptide chain. Okay, so for a polypeptide chain to be called as a protein, at least you should have a number of amino acids, at least around like a 60 amino acid. Okay, so more than 60 amino acid, you will have like a 6 Dalton. If you have like a minimum molecular weight of 6 Dalton for a polypeptide chain, you can call it as a protein. Okay, so less than 5 Dalton, okay, less than 6 Dalton or less than like 5 Dalton, you cannot call that polypeptide as a protein. It will be simply a polypeptide chain. Okay, it's our peptide chain. That's all. So in order to call a polypeptide as a protein, you need at least like 6,000 Daltons. Okay. So you have like a different kinds of like amino acid, alanine, serine, glycine, cysteine, valine. Okay, so you have like a variety of like 20 different types of amino acid. So any combination can be there. There is no rule like after alanine only serine should come. After alanine only glycine should come. Okay, there is no such rule. So any combination you can have to make a polypeptide. Okay, so you can see like it's making a condensation reaction here and condensation here and here and here. So everywhere between like each 2 amino acids there is a condensation and resulting like C double bond O NH. Okay, so peptide bond, peptide linkage, it is making between like each amino acid and resulting a polypeptide. Okay, so if you are making a line, something like this, the red line indicates the continuation of polypeptide. Okay, if this is the N terminal, you will have NH2. Okay, and this is, if this is the C terminal, you will have C double bond O and OH. Okay, but it is continuous polypeptide, so it is it is showing as a broken line. All right. So this is like is our side chain, different R group comes. Okay, so like overall like idea of like a polypeptide chain. Okay, so in the beginning you will have NH2. Okay, at the second, from the second position you will have only NH. Okay, so everywhere you will have only NH except the N-terminal amino acid. And similarly, everywhere you will have only C double bond O. Only at C terminal you will have C double bond O and OH, single bond OH. Okay, so which is indicating like amino terminal and carboxy terminal. Okay, so this overall a sketch of like protein or like polypeptide chain. Okay, so I told you that it is making a dipeptide bond by condensing like two amino acids. By condensing, you are making a dipeptide bond where you will have like a C double bond O connected with amino group. Okay, NH group. It's just like a single bond, C single bond NH. Okay. So here the carboxyl oxygen, it is making a partial negative charge, and the nitrogen also will have a partial positive charge. So there is a small electric dipole present in the peptide bond. Okay, and another thing is the carboxy group and this NH group always facing opposite, or you can say they are always in trans configuration. Okay, the C double bond O always in trans position to the hydrogen of this NH group. Okay, so that means it will be in 180 degree. Okay, only in case of like a beta term. Okay, we will see what is mean by beta term in following classes. So in that case, it will not maintain trans configuration. Okay, so the carboxyl group and NH group will be facing like same side. Okay, so that bond is called cis configuration. Okay, so only in exceptional case, okay, you will have like a cis peptide bond. Otherwise, you always get trans peptide bond only. All right. So trans peptide bond means, so all these like four atoms, that is oxygen, carbon, nitrogen, and hydrogen, they will be lying in same plane. So all the four atoms will be making a planar structure. Okay, so you have like a C alpha atom here, and this side you have like a C alpha atom here. Okay, so here C alpha to C prime, the carboxyl group, C prime, you will have like freedom of rotation. N to C alpha, you have like a freedom of rotation. So this bond you can make a rotation, and this bond you can have a rotation. Whereas this bond you cannot have freedom of rotation. It's a very rigid structure. Okay, so rigid structure means, so it should have the double bond nature, right? If it is a single bond, then you should be able to rotate freely. But since it is making like a double bond character, okay, it is not freedom to rotating. So it will be a rigid structure. So note down the bond length here, it's very important. Okay, So you can see here, like C alpha atom, C prime, and oxygen. And then this is the nitro group, okay, and then H, NH. The next amino acid C alpha, it is coming over here. So you can see the C double bond O, the bond length is 1.24 angstrom unit. Okay, so this is a double bond. The 1.24 angstrom unit is the length of double bond between carbon and oxygen. If it is C single bond oxygen, the bond length will be little higher than 1.24. And then C2C alpha, it is single bond. So the bond length will be 1.54 angstrom unit. Okay, so some people writing like 1.51, but it is not correct. Okay, it's 1.54 angstrom unit. Okay, and then nitrogen and hydrogen, it's atomic distance, 1 angstrom distance. All right. Whereas if you note on this, C prime and N, it is saying 1.32 angstrom unit. Okay, so this distance does not belongs to double bond or single bond. Okay, so it is not the distance, 1.32 is not a distance belonging to C single bond N or C double bond N. Okay, it is in between. So that means it is showing partial double bond character. Okay, so it is a partial double bond, and that's why you are not able to freely rotate along this bond. Okay, carbon C prime to nitrogen, you have restricted rotation. So as a result, this peptide unit will be always in planar configuration, and the carboxyl group and the amide group will be always in trans configuration. Is it clear? Students?
Yes, sir.
Yes, sir.
Yes, sir.
Okay. So you have to know about like a dihedral angle. So what is the dihedral angle? So, in order to measure angle, you require like a 3 points or a plane you require. Okay. So if I have like a 3 points like this, okay, 1, 2, 3. Okay, I can measure the angle between this, because these 3 atoms are like 3 points making a plane, right? So it's making a triangle, it's making like a plane. So I can measure the angle in between this. Okay, similarly, assume like these 3 atoms also making another plane. Okay, so I'm numbering like 1, 2, 3, and 4. Okay, 1, 2, 3 making one plane, 2, 3, 4 making another plane. So I can measure the angle here. So I have like 2 different planes, okay, 2 different angles. Okay, so I have a plane like this and I have another plane like this. Okay, if I measure the angle between these 2 planes, you got it? Students? Say yes or no?
Yes, sir.
Okay, so if I measure the angle between these 2 planes, okay, that is called like a dihedral angle, or I can say it's a torsion angle. Okay. So assume like this as like one plane, okay. Okay, I cannot change the color. So think like, oh my God, this plane is a little bit awkward. Okay, so this is, okay, you have like a one plane here, you have like another plane here. So the angle of this plane you can measure here, and the angle you can measure for this plane. Okay, so you have like two different planes. If I measure the angle between these two planes, okay, so this is called like a dihedral angle or torsion angle. Okay, so the angle between two planes is called like a torsion angle. What is the use of torsion angle? So we will see that. Okay. So since you have like a rigid conformation between these like four atoms, you have like a rigid structure among these four atoms, right? So it will be like a planar. Okay. So you will have like a freedom of rotation over this bond, and you will have freedom of rotation over this bond. So N2C alpha, you can have like a free rotation, and C alpha to C, you will have another like freedom of rotation. So, so this will be acting like a sandwich. Okay. So you can see here, so the angle N to C alpha, it is measured as a phi angle. The torsion angle, it is measured like a phi angle. C alpha to C prime, it is psi angle. Okay. And then C to C prime to N, that is omega angle. Okay. So the omega angle, it will be always 180 degree. Okay. For transpeptide. Okay. For cis peptide, it will not be 180 degree, it will be 0 degree or something else. Okay. So you have like a planar unit over here, another planar unit over here. So you have like a two poly, like this peptide plane, you have like two peptide planes. So between these two peptides, one C alpha is there. So it's like a sandwich. So each residue is sandwiched between two rigid planes. Okay. So can you see this? Okay. You have one plane here, another plane, peptide bond here, another peptide bond here. Okay. So you have like a different amino acid. Okay. So different side chain and or like different amino acid here. Okay. So so you have like a 5 over here. So N2C alpha, this N2C alpha, like you have like a 5, and C alpha to C prime. Okay, that is psi. Okay. So you can have like a rotation over here, or you can have like some restricted rotation between these two planes. Okay. So that is why you are getting like a different kinds of three-dimensional structure. All right. So I have like a different phi, and I can have like different phi angle, different psi angle, which is making like a different three-dimensional structure. So what is the possibility of like making phi and psi angles when I make a different polypeptide chain? So that is what described by Ramachandran Plot. Okay, so you have like 100 and minus 180 to like plus 180, that is like the phi angle on one axis, and minus 180 to plus 180 in like another angle. Okay, that is psi. Okay, so make the like a plot over here, phi and psi. Okay, so you have to make like phi and psi. So both you have, right? So the phi and psi combination, different combination makes different three-dimensional structure. Okay, so in this plot, okay, you can have like any phi angle, you can have any psi angle. Okay, so only allowed region is shown in shaded region here in yellow color and in red color. Okay, so the red region is a highest like confidence. So almost all the possible combination will be found. Okay, only here. So favored conformation, 5-side favored conformation. The yellow region is showing the extended conformation, little bit extended. All right. So you cannot have like more extension. If you keep on rotating, what will happen? So here you have like a phi, you have like a psi angle. All right. So you can rotate this phi and psi only to certain extent. If you keep on rotating, what will happen? So this R, right? You have the bulkier group over here, and you will have like a carboxyl group over here or somewhere here. Okay. So if you keep on rotating, there will be steric interaction between this R and oxygen. Okay. Or if you rotate over here, this R and the oxygen may have some steric classes. Okay. Or if you keep on rotating, this R, the next residue, may be coming closer to this R. All right. In three-dimensional space, they will be coming closer, which is not a favorable conformation. There will be steric class. Okay. So the polypeptide chain will not be stable. All right. So to which extent that you can rotate this phi and psi angle. So that is what described by Ramachandran plot. So in the Ramachandran plot, in the first quadrant, okay, so almost all the beta sheet, okay, almost all the beta sheet will be found only in this region. Okay, so the extended beta sheet you can find out somewhere in this yellow shaded region. And then right-handed alpha helix will be found only in this phi psi angle, the red one, the red color shaded one. The yellow is extended region. And the left-handed alpha helix you can find out in second quadrant, and you will not be able to see like any amino acid, all right, any phi psi combination in the fourth quadrant except glycine. Okay, because glycine does not have any side chain, right? You have only hydrogen group. So you can find out the glycine anywhere in the Ramachandran plot, but not the other amino acid. All right. So if you see like any amino acid except glycine, any other amino acid, if you see anywhere in the Ramachandran plot, in the like non-shaded region, in the white colored region, that is appearing in disallowed region. Okay, so then that means that amino acid you have modeled in a different conformation, which saying there is some steric class in that model. All right. So then what you have to do? You have to go to the model, you have to correct the conformation, and you have to validate again with the Ramachandran plot. Okay. Until you are getting the phi psi angle, okay, in the allowed region. Okay. All right. So this is, I told you, like a general, like a beta sheet. Okay. So beta sheet, you have like a different types of beta sheet is there, like parallel beta sheet, anti-parallel beta sheet, twisted beta sheet is there. Each and every type of structure, secondary structures having its own phi psi angle. You can see here like the phi angle is like around like minus 120, and it is around somewhere like maybe 100. Okay, psi angle. So minus 120 and psi will be 100. So that will be parallel beta sheet. Okay. So then phi will be somewhere like minus 100 for anti-parallel beta sheet, and then somewhere like 130 or 40 psi angle will be for anti-parallel beta sheet. Okay. Similarly for like twisted beta sheet, somewhere around like maybe 100, right? Something 100, and here 150 somewhat. Okay. So here for left-handed alpha helix, you can see here around like 60, right? Phi angle is 60, and here maybe like 50. Okay, so different combination, it's making like a different secondary structure. Okay, so this is actually found, the Ramachandran plot was like introduced by G.N. Ramachandran. Okay, so he was a professor. Initially, he was working in Madras University in physics department. Okay, so he is the founder of crystallography department. Okay, in Madras University, he founded the department, biophysics and crystallography. Okay, so then later on he moved to Bangalore, IISc, Indian Institute of Science. Okay, there also he established crystallographic department. Still, the department is there in the name of Molecular Biophysics Unit. Okay, MBU. Okay, so he only like made this Ramachandran plot. Okay, and who are like solving the structure, whether by crystallography or NMR or in like molecular modeling, whatever. Okay, anybody like making a model for a peptide chain, okay, that has to be validated by Ramachandran plot. So that if the Ramachandran plot says all the amino acids in the model are found lying in allowed region, then the model is correct. Okay, if it is saying no, some amino acids are found in like a disallowed region, then the model is wrong. Okay, you have to remodel it. Okay, so that is like a very good work of like G.N. Ramachandran. So not only that, he had worked on collagen structure. Okay, so when he was analyzing the collagen structure, he invented this Ramachandran plot. Okay, so all these are like a history, but you have to remember this. Okay, all right. So here you can see like different combination. So wait a second, what's this? So here you can see like minus 140, around 140, minus 40 and plus 135. 5 psi angle appearing for anti-parallel beta sheet. Okay, so for parallel beta sheet, it's around like 120, minus 120 and plus 113. Okay, so the right-handed alpha helix is coming around like plus 64 and plus 40. Okay, and then there are like a 310 helix. Okay, and then pi helix. Okay, so different secondary structure which is having like a different like phi and psi value. Okay, so here I told you glycine doesn't have like any limitation. Okay, and it can appear anywhere. And the proline, you know, the proline, it is the side chain, right? So the side chain, the R group is connected with C alpha, right? So it will make like a cyclic structure. Okay, so it's, since it is making like a ring kind of structure, you have to be like very careful of like phi psi angle of proline, right? Okay, so the C alpha atom, okay, the dihedral angle of like the C alpha atom like makes like three different three-dimensional structure, different conformation for protein structure. Okay, so the omega angle in cis and trans peptides. Okay, so here this is the trans peptide, right? So you can see here C prime, double bond O, this oxygen, and this NH. This is the peptide unit here. This is a trans peptide because oxygen and nitrogen, they are the oxygen and hydrogen, they are in trans position. Okay, that is 180 degree, right? Whereas here this is in cis configuration, C prime and oxygen NH, because the oxygen and hydrogen are in same side, they are not in opposite side, right? They are not opposite, they are same side. So this is a cis peptide. Okay. So always trans peptide will have 180 degree. In cis peptide, you will have like a 0 degree or plus or minus 30 degree. Okay. So cis peptides are energetically unfavorable. Okay. So it is like 1000 times less stable than trans configuration. Why? You can see here, this is the C alpha atom. You will have R group somewhere here, right? You have the R group somewhere here for this C alpha atom. So this is trans configuration. Okay. So the R group here and here, they are happy, right? Because they are far away from each other. But in cis configuration, the R group is coming here and the second R group is coming here. So what will happen? You will have steric clash between these two R groups, right? So energetically, this will be less stable, 1000 times less stable than the trans configuration. So most probably in almost all the protein structure, you will always have like a trans configuration only. Okay. So there are like exceptional cases. Okay. So where you have like a proline residue, right? So you see this proline residue. This is C alpha atom. Okay, and the side chain that is coming over here, C beta, C gamma, C delta, which is making a bond, covalent bond with the nitrogen. All right. So this oxygen is here. So actually the hydrogen should come over here, but there is no hydrogen because it is making a covalent bond here. All right. So the trans configuration, this is the trans configuration in like a proline peptide bond, and this is cis configuration. So in trans configuration also you will have like a steric hindrance, and in cis configuration also you will have like a steric hindrance. Okay. So whatever it is, the steric hindrance will be there. Okay, just like 4 times lesser than trans peptide. In cis configuration, the energy difference is just like 4 times lesser, whereas in the other peptide is like 1000-fold. Okay. So this like a cis trans isomerization is like very important in certain protein folding. Okay. So the prolyl peptidyl isomerases. Okay. So in case of particularly in case of like this prolyl peptidyl isomerases, this cis trans configuration is like very important. Okay. For during the protein folding session. Okay. And so how far it is allowed in like Ramachandran plot. Okay. So like 2% is generally allowed. Okay. So if your protein model is having like a 2% of amino acid appearing in disallowed region, it's okay. Okay. 2 to around like a 5%. Somewhat like some people is like allowing. But more than like 5%, then there is serious problem in your model. Then you have to remodel your structure, and again you have to validate with Ramachandran plot and bring the value less than 5%. Okay, so then only you can say that your model is correct. All right. So Ramachandran plot not only measuring only like phi and psi angle. Okay, so it will check the conformation for even the side chains. Okay, so for each amino acid you have like a different side chains, right? So even Ramachandran plot measures the side chain angle. Okay, so if the side chain angle is like coming in a disallowed region, then again you have to go to the model and correct it. All right. Okay. So these are like some essential amino acids. So in your food that you have to be careful to take the essential amino acids. Okay, otherwise some nutrition problem will come. Okay, so you can find out all these like essential amino acids only in non-vegetarian. Okay, so the vegetarian people will have like less chance of finding the essential amino acid uh, in vegetables. Okay, so in that case, you have to use like all kinds of vegetables. You have to use like a curd. You have to use like a egg. All right. Otherwise, you will have like a shortage of the essential amino acids. Okay. All right. So complete protein. Okay. So which will provide all the essential amino acids. Okay. I told you already. So meat, fish, milk. All right. So vegetarians should eat like many different kinds of plants or supplement diet with milk and egg. Okay. So there are like some rare amino acids like 4-hydroxyproline, 5-hydroxylysine, which is found in collagen. Okay. So this is like very rare amino acid. And then D-glutamic acid. You see, this is D-glutamic acid. So naturally, we have L amino acid only. But in cell walls of bacteria, you have like a D-glutamic acid. In earthworm, you have like a D-serine. And gamma aminobutyric acid in neurotransmitter, and beta-alanine in like vitamin. Okay. Okay. So with this, I stop. Okay, so we will have a smaller break here, and then after like some break, we will again continue our class. All right.